Good afternoon and good morning or good evening, depending on where you are. My name is Enrique, and I'm one of the parents and researchers that collaborate here at the Autism Research Coalition. Today is a very special day because we have a very, very special guest with us, Dr. Juergen Hahn. Dr. Hahn, thank you very much for joining us.
Well, thank you for having me.
So Dr. Hahn has a very impressive background, and I'm going to ask him to introduce himself in just a moment. But I would like to give folks a quick 30,000-foot view of the importance of having this live chat with you, Doctor, if you don't mind. So Dr. Hahn and his team are the scientists and also act as a data scientists behind some very, very important research that has emerged in the autism work just recently. This research includes a collaboration with the Arizona State University team where Dr. Hahn conducted a very specific analysis with the data from this now famous Microbiota Transfer Therapy trial led by Dr. James Adams. So Dr. Hahn has also led several other multivariate analysis with data from clinical trials and also with big data with one specific objective, which is to study the differences between children with autism and neurotypical children. And perhaps even more critical is the fact that Dr. Hahn and his team are currently in the process of developing a much needed diagnostics tool for autism, which to my knowledge would be the first clinical, as in non-behavioral, way of diagnosing autism and to evaluate progress or gains, as far as usually call them, as a result of biomedical treatment. So all of this is very, very exciting and very critical research, not only for people diagnosed with autism, but also hopefully will become a tool to help diagnose children with autism much earlier in life, and maybe even looking at potential biomarkers for women who may be at a high risk or of having children on the spectrum. And so we're going to talk about, these different things today. So I think that the last thing I just want to, mention is that for us, these are not, is very, very important research because the autism is really a pandemic and it's incredibly alarming. As we all know, according to the CDC, 1 in 54 children in the US is diagnosed with autism, and it is 1 in 34 among boys. And there are also reports of even higher rates in other countries like Ireland or Northern Ireland, when 1 in 24 children is diagnosed with autism and 1 in 16 among boys. So again, super important and very appreciative of your time today, Doctor. So why don't you go ahead and introduce yourself and your professional background, your motivation for doing research in autism?
Okay, well, thank you for the introduction. So I'm a professor at Rensselaer Polytechnic Institute in the Department of Biomedical Engineering, and I also serve as the department head of that department. Yeah. Basically, I mean, my background is for the last 20 years, I've really been working on what I would call computational systems biology, yeah, where nowadays people would say, oh, this is biomedical data science, since everybody likes talking about data science now, but we really worked in this field long before these terms became fashionable. Yeah. And so traditionally, we've done a lot of work on trying to gain better understanding of what is really underlying certain diseases and conditions. And we've had looked at things like inflammatory bowel disease and so on for the longest period of time. Yeah. And then now for the last 5 years, we really translated all the algorithms and the techniques that we've developed into the autism space. Yeah. And I mean, part of the motivation for it was simply, if you had looked at the literature at the time, yeah, I found that the level of data science that was applied when it came to autism was clearly not where other fields in engineering and medicine are. And so I saw that as a tremendous opportunity and a need also to basically come up with to basically use our techniques in order to gain a better understanding of what's going on really when it comes to autism. Yeah, so that was a big motivation for us there. And what the other thing, I mean, related to that, to just give you some ideas, that if you go back 10, 20 years, people in general, like they were looking at, is there an autism gene, or is there one biomarker that you can find? And the reality is all of these ideas came up empty. Yeah, I mean, we're talking about when they are even on the genetic side, people are talking about dozens or hundreds of genes, and the fact that they only raise the risk a little bit, so it's not like that you can usually use them to predict. Even with other biomarkers, there is not one that really tells you what's going on. Yeah, and this is where our work comes in, because really what we've always been doing is that we did what I would call multivariate analysis. So we look for, are there patterns in the data? Like if I have one measurement that's a little high and it's of some quantity, and a second one that's a little low, and a third one that's a little high, then each individually may not tell me much, but together they can paint a very clear picture. Yeah, and that is something that I was really missing when I looked at the literature related to clinical studies for autism spectrum disorder. And so this is how we got started with this. Yeah, and one thing that's really important for us is, I mean, I come at this from the data space. Yeah, so I don't run clinical trials myself. I work with people who do. And so I needed good, strong collaborators in this field who run a lot of clinical studies. And luckily, I was able to find some very productive collaborators, and that's how everything got started. Yeah, and so for the last 5 years, now my group is almost exclusively focusing right now on different aspects related to autism spectrum disorder. The diagnosis part is a really big chunk of that, but we've also done some work on, for example, how do you if you take measurements from mothers during pregnancy, can you predict something about the risk? We've done some work related to if you run a clinical trial, can you use some of these? I would call them biomarkers, but they're really combinations of measurements. Could you use some of these biomarkers to actually predict the success of a clinical trial, or not? In addition to just the observations. We've done some epidemiology studies related to what are factors that may or may not increase the risk of somebody being diagnosed with autism later on. So this is kind of in a nutshell what my group is looking at in a variety of different ways.
Thank you for that introduction, Doctor. I would like us to start with a piece of research that is near and dear to the heart of many people in the autism world today, because it has given a lot of hope to families, and it is the now famous Microbiota Transfer Therapy clinical trial led by Dr. James Adams and his team at the Arizona State University. For those people that are not familiar with the trial, I would just kind of, I guess it would be probably sufficient to say that these MTT, as it is called, Microbiota Transfer Therapy, has been without question the most effective treatment in reducing autism symptoms that has been ever, recorded in a clinical setting. For the average kid in the trial, the benefit of this treatment was in basically cutting their autism symptoms by half 2 years after the MTT. So that was pretty remarkable. You, Doctor, you conducted some analysis based on these data. So would you mind describing the analysis that you did, what kinds of patterns you were looking for, and what were the results of your analysis?
Okay. Yeah, and so one of the things that we're trying to understand, and please understand that this is really still work in progress, is that the microbiome is a very, very complicated, is a very complicated subject, in that you have hundreds and hundreds of microbes that serve for certain functions, and they basically, at some level, they support each other, and so there's many interactions going on, And what we tried to do with our work was basically taking this data, both data taken at baseline, basically when the trial was started, comparing it to data from typically developing kids, and then tracking how did the microbiome change over to the course of the trial as well as after the trial concluded. Okay, what we want to get at is not only can you see that when we looked at the plasma metabolites, that they became closer to the typically developing cohort as the trial went on, and what you're seeing there, the top one, this is basically at baseline, and then as you go down, you see how this basically evolves over the period of the trial, and what we want to get at is so first of all, showing that yes, this becomes much closer to what the measurements for the typically developing cohort look like. But also you really have to distill this data of many, many different measurements down into a few measurements that we can use like in a figure. Okay, and that's really where data science helps with visualizing these very complex, phenomena that we're seeing here. Yeah, like for example, like if you look at their figure A, the one at the top, in red, this is the basically information from the children with ASD before the trial started. Yeah, what you see in blue, that's the information from the typically developing cohort. That's in part A. Then what you basically see in figure B is that now this is the information from the ASD cohort after week 3, so after basically the trial has been going on. And you can see that this has shifted. Like if you look at this dashed line, and I mean, you can put the dashed line more or less wherever you want, determine there's a separation between the two groups. But you see now a lot more of the yellow curve is to the right of the dashed line, so closer to what you would expect for the typically developing group. And then when you look at the figure C at the bottom with this pink line, you see that it has shifted even further towards that group. Yeah, and so basically our work is able to visualize how you see these changes. Now in this case, in the plasma metabolites in response to MTT. And then we also wanted to know, so what are the key metabolites that really are changed in this? Because if you want to tailor your strategies at some point in the future to figure out what are the key variables I need to have an influence on, we first have to identify what those are. And that's always the difficult task. I mean, part of the problem with these things is you have so many different measurements that it's kind of, you can get lost in all the measurements there. And so our work helps visualize this and show what are really the things we want to focus on, as well as tracking progress.
Right. This is I think incredibly remarkable. I mean, that we can see a progress like this. This is I mean, from my perspective, you know, I'm personally just in full disclosure, I'm also a data scientist. I mean, to see this kind of you know, trends as time goes by, and just to clarify for people again, just that may not be, you know, familiar with the trial, this was a minimally invasive treatment. I mean, it started with a reset of the gut in children. They were given an antibiotic called vancomycin for about 2 weeks, which is, I think that what this week 3 represents. It would be that initial trend after the vancomycin, and then they had a bowel cleanse, and then they had the MTT, this transfer of microbiota, which is kind of a highly purified liquid. That's my understanding, containing the bacteria of healthy and highly screened donors, being transplanted into, you know, either orally or rectally in these children for about 8 weeks. So that's kind of what this timeline represents. So we go from, you know, time 0 on this red, and then we go with vancomycin, and then finally the microbiota transfer therapy. And this kind of score, I suppose that this is a composite metric, right, Doctor? It's kind of like a combination, like as you were referring to, of multiple metabolites that you were assessing, correct?
Yes, that's exactly what it is. And the quick explanation, I think you explained what happened very well. Is they got a large initial dose shortly after the bowel cleanse, and then they had to take a low maintenance dose for a few weeks afterwards. Okay, and so yeah, but you're right. This is a composite score, because the problem you always end up with, if you're just trying to track one measurement that you have, it's really not as easy to see how much progress you're making. Yeah, and that's really, whereas I think in this figure, it's relatively easy to understand that clearly this curve has shifted over to the right side, more to the typically developed, and looks more similar to typically developing.
Yep, and then there's also this chart here where we can see also the progress for each of the individual metabolites, right? As they were, as you know, each measurement was progressing over time, and how they were becoming closer and closer to the typically developing children. So again, this is just a different view at how the multivariate you know, technique is being applied at a composite level, but also at the individual level, how this is also you know, surfacing in a very nice and compelling manner.
Mhm.
I think one question that I have for you, doctor, is we're using here 3 plasma metabolites that may represent like 3 general groups, right? And the level of accuracy or specificity that you found with this approach is pretty remarkable. So I would expect that, you know, this algorithm would be like much more complex than this one. But these metabolite profiles really shifted significantly to become more similar to the ones of the, you know, neurotypical children. And actually in the paper you mentioned that there are a couple of metabolites, that is sarcosine and inosine monophosphate, that really improved greatly, which are the first the first and the third, I think, charts here that we're seeing. So what do you think is the significance of something like this? And also, if you can just kind of clearly, I mean, briefly for the people that are watching us that may not be familiar with the terminology, what is a metabolite, and why did you guys choose to zero in on looking at metabolites as potential treatment for autism?
Well, I mean, so basically, a metabolite is anything in the metabolic pathway, and ultimately, metabolic pathways are really driving cellular processes in your body. I mean, anything that involves energy production, there are metabolites involved. Yeah, and one of the things, now not just for this trial, but in general, that I wanted to focus on with my work is that traditionally, people had usually viewed autism really more from what you may call the psychological side. They see that there's behaviors, and we can measure the behaviors, and you have a diagnosis based upon behaviors. But the reality is there has to be something underlying in the body that is causing these behaviors to emerge over time. Yeah, and so metabolites is one very good way to do this. And let me take a step back there. I mean, there's been a lot of work on genetics related to autism. Yeah, and I really did not want to go down that route. And let me tell you why. Okay, is that one thing is that people like genetics because the genes almost pretty much don't change. Okay, they're the same the day you're born as well as the day you die decades later. Yeah, so if you have a certain condition or not, your genes will not be changed by having this condition. Yeah, what it really comes down to is your genes is more like a probability that you may develop something. Yeah, like a lot of people know that that certain genes, if you're a woman, you have a much higher chance of developing breast cancer later on. That doesn't mean you will. It just means you have a higher chance. Yeah, but you already have these genes when you're a teenager, even though you may not develop this until decades later. So if I want to know what is my condition right now, my genes do not tell me that. I mean, there are some diseases where you can say, okay, you have this gene, you have this disease, but that's not always so simple. I mean, when you look at autism, you have some people that come off the spectrum, and you have other people who meet all the developmental milestones when they're younger, and then they regress and are diagnosed with autism later on. Yeah, so it's not as simple. There may be predispositions there, but it doesn't help me with the diagnosis right now. Okay. Now, metabolites tell you more about a snapshot about what is going on right now in the body. Yeah, they're much more difficult to deal with. This is one of the reasons people don't necessarily like dealing with metabolites. But they give you a much better idea about generally what is going on right now. Okay, and that's why we focused on saying, look, let's look at studies where metabolites are measured, because they give me a better idea of what is happening right now, whereas the genes are more a blueprint of what may happen over the course of a lifetime. Yeah, like if you wanted to, and please interrupt me if you think this is too simple, but if you wanted an analogy,
go.
Yeah, basically, if you were to buy a house, yeah, and the house is 20 years old, the genes are like the blueprint of the house. I can look at the blueprint and saying, oh, this looks all like it's well built, or maybe it looks like, tells me that, oh, there's something, this is built in a weird way that could cause problems in the future. But now that the house is 20 years old, it doesn't tell me what's going on right now. Like maybe the house was struck by lightning, and or maybe they have a hole in the roof because a branch fell on it. the blueprint doesn't tell me that. So I need the current understanding. And so the metabolites is more like I am investigating what's going on right now. Now, metabolites have drawbacks, okay, in that they're not only difficult to deal with, but basically they're influenced by things like diets and so on. Yeah, and so you have to make sure that you have some level of control. Yeah, that we're not analyzing children where the diets are totally different. That ultimately you're tracking the wrong thing as a result. Yeah, so you have to be careful with dealing with it, but it does give you a much better idea about what is currently happening in the body. Okay. So and that's why we focused on metabolites. Now, these particular metabolites, I mean, here's what we generally do. Okay, when we have a study, we try to with our collaborators, obviously, we can try to come up with a large panel of measurements we have. There are some metabolites that if I can measure them, I would say, let's try to measure them because I generally find that they're good indicators of what we're seeing. Okay, I'll give you a few examples. I mean, many children, maybe even most, yeah, they have some first challenges with oxidative stress. And so there are metabolites that are good indicators for oxidative stress. So I would generally say I would like to see some measurements there because that gives me some information. Yeah, but again, it's not universal. Yeah, and but other than that, we try to say, well, if I can have a measurement panel of a few hundred metabolites, why not use it? I mean, nowadays, high throughput technologies are pretty good and they're not that expensive. And then I use our algorithms to determine out of all these measurements, so what are the ones that really matter? Because I don't know going into the study exactly, let's say, should we have measured sarcosine or not? I don't know that. That's what came out of it. Okay. And our algorithms determine basically what is the best combination of measurements from everything that we've taken that basically allows us to find differences between these two groups. Yeah, and our two groups, at least at time 0, were children with autism and typically developing children. Yeah, and so what we did for like this study, we basically looked at all combinations of metabolites, of all combinations of 2 metabolites, of 3, of 4, of 5, yeah, and saying which gives me the best separation. And what's really important here is that people, they look at what you call a training data set to find the best separation, but that's really not fair. What we want is we want the best separation on a testing set. Yeah, and so we use most of the time something called cross-validation, where we only test on data that was not used as part of basically building these models. Yeah, and then if you find out, for example, that let's say sarcosine is one of the variables that tells you the most, okay, now we got one. Yeah, it may also tell you there may be other variables that tell you quite a bit, but that behave in a very similar fashion. That means that these may all be important, but probably I only need to measure one of them because the other ones are correlated. Whereas if I have a set of 2, 3, 4, 5, the set means they complement each other, the best way to show differences. That's really what we're doing. Yeah, and so most of our studies, we have a lot more measurements than we're using in the end to build these classifiers. Yeah. So like the smallest measurement sets we ever had like 24 measurements, and then we have others that go and have 600, 700, 800 of the different properties that we're measuring. It all depends on what's available. Yeah, but in all cases, we have a lot more measurements than we ultimately need, but we're still at the stage where nobody really knows what are the key metabolites. That's what we're trying to establish to determine, well, not are these good predictors, but maybe we can influence some of these predictors in a controlled way in order to hopefully get a desirable result.
Yeah, that's actually a super important point because, you know, as you mentioned, this research doesn't necessarily mean that these are the only 3 metabolites that are important. I mean, like you said, I mean, there may be like, you know, like many more that may be very closely overlapped with the ones that you were using for these discriminant analysis that you did. And actually, I would expect that something like that would happen, and given that you use like 621 metabolites, if I'm not mistaken, in total. So, I mean, all these different combinations, I'm sure you got like some combinations that were actually pretty close. It's just that you decided probably to use, I'm guessing, the one algorithm or the one triplet, so to speak, that gave you the highest level of discrimination between the two groups, which is, in and of itself, is pretty remarkable, like I said. But it's good to clarify that this is by no means the only three that are important. I think that one of the questions that I have, Doctor, is there are many kinds of metabolites. That's my understanding. There are primary, secondary metabolites. Is there any reason or any rationale behind the selection of the metabolites, or you went like, as broad as you could go and simply choose whatever metabolites you could use, or was there any screening criteria for that?
It's a mix, okay? I mean, one thing is we wanted reasonably broad, because look, we know metabolites are usually part of one or multiple different pathways, and these pathways are tied to direct functions in the body, yeah? And sometimes they're tied to multiple functions, unfortunately, and it gets more complicated. But so we want to make sure that we have a broad spectrum to cover many different metabolic pathways. Yeah, I mean, as a minimum, at least two, but hopefully more. And like in this study, we had a bunch more. Yeah, and so that, because it gives us a better idea if maybe there are certain pathways that are more perturbed, yeah, in children with autism than others, because that gives you a better understanding of if you really want to design an intervention where you look at. Yeah, I know, like, we can talk about this later, but the work we've done on the ASD diagnosis, that actually started with the idea that we wanted two pathways to focus on. And then we picked metabolites from these two pathways, not knowing which were the best ones, but then we had a much more, I would say, much more targeted approach and much fewer measurements. Yeah. In this case, given that the microbiome is still honestly, we're in the infancy of understanding how the microbiome really works, not just for autism, but in general. Yeah. We wanted to say, let's keep it broad because we really have to get gain some better insight. But we certainly wanted to make sure it doesn't help me if I have 40 or 50 metabolites from one pathway instead of 10 or 20. Yeah, that may not add a lot of information, but it is more important for me that I have a number of metabolites from a number of different pathways telling me, oh, well, it seems like this type of metabolism is perturbed and this type of metabolism is perturbed due to this condition.
Yep. Yeah, that's a great point. So I guess I'm just kind of to understand the real significance of some metabolites specifically, right? So even though there may be like some overlap between these three and others, I would imagine that there's got to be like a relatively small subset of biomarkers or metabolites that could give us some indication of cause and effect, right? I mean, obviously we don't know that at that point, but that's I suppose that's kind of what the research that you're trying to get at. I mean, in defining these diagnostic tool is getting towards.
Oh, absolutely. So let me shift gears a little bit to our diagnostic study, which happened before this. Yeah, and because there. Exactly what you're measuring were the thoughts that went into this. Okay, and so what we were interested in, and I mean, this was published 3 years ago in PLOS Computational Biology. Yeah, I mean, we know that a lot of children with autism have problems with oxidative stress, and so we said, fine, the body's most powerful antioxidant is glutathione. Yeah, so let's look at a pathway that leads to the production of glutathione. Yeah, and see if there are perturbations in that pathway. And the second thing we wanted to look at is, I mean, there's basically a lot of talk about that, well, maybe the epigenetics in children with autism is different from the typically developing children. And so we said, well, you know what, there is a pathway called the 1-folate carbon metabolism that regulates DNA methylation, which is the regulator for the epigenetics. Okay, so if the regulator is off, then the epigenetics will be off. And this pathway actually intersects at some level with what's called the transsulfuration pathway leading me to glutathione. So I had two pathways. Now, in this case, there were two pathways I was interested in because there were plenty of reports that you see differences there, but they're also interconnected, connected at some level. And so we said we picked a good enough number of metabolites on both of them, and then you basically determine from that what does this tell me with differences between the children with autism and those without? Yeah, and we found, I mean, for that case, if we use this as a diagnostic tools, we had like sensitivities and specificities that were like 96 and 97% in that range. Yeah, so really, really high, and we had a good number of participants. It was over 150. Yeah, so it was a big study, and but the reason I mentioned that is there we had some understanding. They say, look, oxidative stress is reported oftentimes in the literature, so it's likely that that plays a role. So we focused on an antioxidant because that is basically, I mean, if you have oxidative stress, the antioxidant has to be dysregulated in some form, yeah, or there's too much oxidative stress on the body in general. And so basically, we focused from what has been reported down to particular metabolites, and then measured metabolites within these pathways. Yeah. Now, a lot of studies that people had done related to supplementations, they are tied to these pathways. Like if you look at anything related to methyl B12 treatments, methylcobalamin, folinic acid. I mean, the folate certainly plays a role. All of this, the pathway is affected by this. And so we said, well, then you have an idea, not only may there be differences, but this could also somewhat explain why by certain interventions may be more successful than others. What we did there for diagnosis was not that we said, oh, all children have this and this is the cause of autism. We don't know what the cause is. Yeah, and but we said that clearly this is one thing that seems to be different, because when you look at diagnosis, I just need to know this is one aspect that children with autism have differently, and you may find that behaviors may improve if you can basically if you can better regulate this one particular aspect. But that doesn't mean that basically people are suddenly off the spectrum if you could do that. But that was never the point. The point was just saying we need a correlation. For diagnosis, a correlation is good enough. Yeah. Now coming back to the microbiome studies, again, those are much more difficult in the sense because we're really just trying to understand the microbiome, and that adds another layer of complexity where we have to do this work now that hopefully in 5 or 10 years, we have a much better idea of saying, okay, this is how the microbiome functions, and this is how it functions differently in children with autism, in order to figure out what works, what doesn't. I mean, like if you look at, for example, the studies there by Jim Adams and Rosa Kralmanik-Brown, I mean, they are taking a microbiome from a healthy donor and saying we try to essentially replicate that in the children. Yeah, and it's been very successful. But they're not trying to tailor this for an individual at the moment because we don't know how to do that. Yeah, in the future, at some point, we may be able to do that. But right now, I think those results are very, very good for what they are already. Yeah, and I think that's a very nice, promising step. But it's clearly in the long run, we want to be able to really figure out what makes a healthy donor, what makes one that would work particularly well for an individual. But we're really not there yet.
Yeah, there are a couple of things you said, Doctor, which are I think very important to highlight, right? So there may be other interventions, right? Like supplements, like you mentioned, or diets, and that actually kind of provides a rationale for people to understand that it's not only microbiota transfer therapy. There may be other things that may alter or influence the metabolites that people can start doing right now. Like correcting the diet or applying the right supplements and those kinds of things to kind of cause some, benefits. Obviously, they're not going to be to the extent of a biomedical treatment, but I think many people sometimes have these questions as to whether or not the diets actually work or if the supplements actually work. Well, there may be some of those things that could be beneficial for certain kids, right?
Yeah, look, one size certainly does not fit all. Okay, that's all. But that clearly there is not one intervention that will work for everyone. That's clear. Yeah, and there may be certain things that may work for almost nobody. I don't know, and there are others that seem to work for a reasonable enough percentage that maybe it's worth trying. Yeah, but that's not I mean, for what it's worth, that's not really for me to answer much. Yeah, that's really, you have to work with your doctor on that. We have done some work on where we, for example, compared 3 different, what you may call relatively or dietary treatments. Yeah, and those were related to basically making changes to the folate 1 carbon metabolism that our diagnosis was based upon. And some of them were more successful than others. Yeah, but none of them worked for all of the kids because, again, everyone's different. But some of this may be worth trying. It really depends. I mean, it's not something there where I can really give a recommendation. And honestly, I'm not qualified to do that either. You need to really, that's what the MD needs to do, not a scientific researcher just as myself. Same thing, I mean, with even with like microbiota transfer therapy, is that a lot of kids have GI issues if they have autism, but it may not be 100%. Also, the percentage seems to be reasonably high. The study you're focusing on, it had to have the criterion that you have to have autism and reported GI issues. That was a criterion in order to be able to participate. So, for example, we don't know what would have happened if you have autism, but no reported GI issues. Maybe the results would not be as good. We don't know. It wasn't part of it. So the reality is, I mean, the human body is very complex and no one size fits all. But that's, for what it's worth, I don't even think that's just for autism. That's just in general something that what works for some people may not work for others. And that's always important to keep in mind.
Yeah, just before I move to some questions about the diagnostics tool specifically, I'm just curious. I mean, there is this other publication that you participated on that looks at specifically metabolites that come specifically from microbial metabolites that come from feces specifically, and it seems like the connection with the other work that you did on plasma metabolites is kind of similar, at least the approach or the process that you took, but were there any differences in the way that you were able to predict or describe or find connections between the metabolites from coming from plasma or coming from these microbial metabolites from feces?
Well, that's, I mean, this is very difficult. Question is very difficult to answer. Yeah, because there is a lot going on. I mean, at some level, the microbial metabolites, they regulate themselves as well as there's interaction with the host, meaning you, the human body. And but that doesn't necessarily mean that it's so simple as this. If one metabolite is high in the microbiome, that it's also going to be high in the plasma of the person. It may just basically influence certain other processes in the body, and you see changes here and there. What we found, but this is really this is not published yet, but what we found is that for this study, our plasma metabolites were more predictive than the microbiome metabolites. That's a stronger, we're not 100% sure why that was the case. I mean, maybe it's because, there's strong regulation of metabolites in the body, and that minor changes from this regulation show up as clearly as, but we honestly don't really know. This is more a hypothesis than anything. But we've looked at both of this, but what it comes down to is there's very complex interactions between really the microbiome and the human body. And as you know, the immune system plays a key role in interacting between the microbiome and the human body. And so some of this may be that you're looking at factors from how the immune system gets modulated by the microbiome at some level. So it's really not easy to draw clear-cut conclusions from it.
Yes. No, makes sense. I don't know if you would be able to answer this question. I understand all the the complexity of what we're dealing with here, but I guess the million-dollar question, I guess, is whether or not it would be possible at some point, given what we know now, to somehow produce these kinds of changes that we have seen using MTT, for example, in the metabolism of children with autism by other means. Let's say that is not a full-blown microbiota transfer therapy, maybe a treatment that only uses, let's say, certain let's say, certain bacteria species or a relatively few set of bacterial species, or maybe even, a treatment that doesn't involve gut bacteria and that somehow is able to also modify these key metabolites that, in the same way that it happened with MTT, or maybe even better. But do you have any insights on that?
This is, look, this is exactly where people want to go in the long run. Yeah, that's basically, if you know what are the key species and ultimately key metabolites you want to influence that these species influence, then you could supplement, with just what's needed. Supplement really, in the term of some form of probiotics or so. Yeah, we're not there. Yeah, yet, but ultimately that's where you want to go in the long run, because we have to better characterize really the microbiome and what it does. Yeah, but you're spot on. The hope is that yes, in the long run we can design these types of therapies which really would then be tailored to an individual. They can say, well, you're low on this, and you're low on this, and actually in this area you're reasonable. We don't need to do anything about it. Rather than just saying, well, let's try to eradicate everything using vancomycin and then try and repopulate the human gut. That's the current approach. But in the long run, that's the plan where people would like to go. But again, that's a while off. You have to run the trials and see how it works. We're really not there yet.
Yeah, I have a question from people watching. So this is from one of the very knowledgeable parents from the autism groups. His name is Ted Hu. He says, any insights on how methylation markers relate to metal excretion and biometabolites?
Yes, look, this is basically there's a correlation there, because ultimately if you want to have, for metal excretion, you're going to need some, basically glutathione plays a role in that. Yeah, and so your oxidative stress, like getting rid of waste or harmful products, is part of, can increase oxidative stress. Yeah, and so that was one of the reasons we were interested in looking at glutathione. What I would want to be careful about is that you are seeing there are some differences in the metal excretion profiles in children with autism versus the one that don't have autism. But my feeling is that this is actually more a result of the methylation working differently, meaning the metabolism, rather than other things. Yeah, like there's people often talk about saying like, well, yeah, that maybe the amount of metals is very different. I think it may be more related to the methylation, the pathways working differently. That's the differences that we're seeing, which ultimately can lead to accumulation of metals. But that's where it gets difficult. Yeah, but clearly, glutathione plays a role in this, and so does the transsulfuration pathway. But if I want to make a guess here, I think that really it's actually the abnormalities in the pathway that you're seeing that leads to these differences.
Thank you for that. So I would like to just ask a couple things about your latest publication, and I think it was released earlier this week, if I'm not mistaken. The one about these diagnostics tool. Is it recent? Because it sounded like it was something that happened a little bit before.
Well, okay. So the first paper that came out about the diagnostic tool, that was about 3 years ago. Yeah. But it's work in progress. We've had a very nice study and analysis of the study showing that there are strong differences. That basically, if you give me a blood sample and we measure a certain panel of metabolites, then if I pick 5 of these metabolites, I can predict with high accuracy if the sample came from a kid that had autism or did not. Yeah. But there's more work that needs to be done there. Like, for example, one thing people were concerned about is that so when you determine, like, does the sample come from somebody either who has autism or who has no autism diagnosis, where do you put the cutoff between which of the two groups fall into? Yeah, and initially we put the cutoff somewhere in the middle and saying, well, we'll get some wrong on both cases, but as long as I get like 97% right, That's a very high percentage. I'm not too worried about. Okay. The criticism is that saying, well, if you were to test everybody, yeah, then and let's say you have 2% of the population has autism, which is reasonably close to the number that you have in the US right now for 8-year-olds. Then having 3% wrong, that means you just have more false positives than true positives. I agree with that, but we were not really thinking about having a test where you would test everybody. We were just wanted to show that this is real. But all you can do is you can change the cutoff. Okay, I can change the cutoff so that I have basically, at least for the samples that we had, that I have almost 100% accuracy when I predict if somebody has autism. But I'm going to miss a few that are falsely classified as not having autism, who may have to come back later on. Yeah, and so we were showing that what happens when we switch the cutoffs. And so the paper you're mentioning, this was actually a review paper, yeah, where we looked at many other. We wanted to basically make a survey of what's the state of the art in this field right now. Yeah, and we focused not just on metabolic markers in the survey, but also on other techniques from imaging and so on. Yeah. But then we basically also, when we talked about the metabolic markers in this, we thought that there were a few loose ends we need to tie up. Now, one was to show that if I change this cutoff, then you do not have this problem of a lot of false positives. That really was just because we picked a cutoff that was somewhere in the middle. And as silly as it sounds, but it takes me about 2 minutes to change the cutoff. It's really not that difficult to do. Yeah, and but people wanted to see the numbers, what would happen. Okay. The other thing we wanted to discuss in more detail is, I'm not convinced that you will ever have a test where you can test everybody, especially at a very young age, and then says, oh, you're going to have an autism diagnosis later on. Yeah, I mean, there's a lot of heterogeneity among the kids that have autism. So we figured out, like, well, we need to discuss a little bit. It's much more likely that as somebody who were to, if you had a test, it's much more likely that this would be administered by or for children who already there are concerns, yeah, because maybe they're missing developmental milestones. So if the main people that you're trying to compare are children that have developmental delays, then you need to compare children with developmental delays that have autism versus children that have developmental delays and have no autism. So that's a very different clinical question. But for that, you need data for children with developmental delays. And so our recommendation was to discuss this, saying maybe somebody should run a trial on that. Yeah, and that's something we'll hope we'll be able to do that with some of our collaborators in the future. Yeah, but it was basically to talk about there are different applications for this, because it's like many times people just think about, you have a test, you test everybody, and then you're going to draw some conclusions from that, and you're good. I don't think this is going to be the case here. Yeah? It's much more likely that you focus on children where there are concerns, and because if there are no concerns, then yeah, I mean, that's you may not find anything. The other problem is nobody's run these type of analysis on 6 months old. Yeah, you don't know what you're going to get. We don't know either. If I don't have data from 6 months old, I can't draw any conclusions from it. Yeah, or 12 months old. And so, so this is really still work that needs to be done. But we wanted to point out what are some of the future points that people need to focus on, as well as what we've developed, how this fits in there. Yeah, for example, another work paper, what we're currently working on is, we all know that children with autism have a lot of co-occurring conditions. Yeah, much more so than children who are typically developing. So the question is, how do these co-occurring conditions, how do they affect test accuracy? It's another thing that we're currently working on right now. Yeah? And so it's these types of things that, so the paper you were referring to that came out very recently, it was this review paper with a much more detailed discussion on where some of this needs to go to tie up some loose ends.
Yeah, I guess I mean, in this review paper, the recent one, you do review various studies that have been done using neuroimaging and brain connectivity and functional MRIs and brain activity, genetic, epigenetic markers, and in addition, of course, to the metabolic markers that we have discussed based on blood and excretory metabolites. And then it also accounts for the prevalence of the autism disorder in the general population. So I guess my question is the goal when developing a diagnostic mechanism of sorts, right? Understanding the complexity of it and how difficult it would be to kind of be 100% applicable to everyone. But is the goal to have an all-inclusive tool that can use all these different metrics, or is it the goal to create a simple tool based on a few markers, like in the case of the plasma metabolites of the MTT trial, that can somewhat reliably diagnose autism, and tying it with a question from Tamara Melkin, how far are we from actually getting a diagnostic tool like this to the general public?
I mean, the goal is you want it as simple as possible. Okay, so you don't want, I mean, like, sure, you can maybe get better accuracy if you run a big spectrum of different methods. But if I can essentially have a test where it's one blood sample with relatively minimal blood draw, where I'm measuring a few metabolites, I mean, ultimately, it's also a cost question, right? You want the cost to be reasonable, because otherwise you can't afford to test as many people as you need to. Goal. And, so how from that, we filed for some intellectual property for what we've done, because basically this use of machine learning techniques on metabolite data, to predict an autism diagnosis, and that's the focus on our IP. The Biorosa technology that is this are planning to they are planning to develop test based upon it. But the first thing is, well, you're going to have to run a larger trial to validate these results in that trial, and you also, as part of that, like, you need to have some kids who have developmental delays in there. That needs to be part of the trial. Yeah, you also need to make sure is that what we've done is that like the measurements that were used to get these metabolite concentrations, they're nice to do in the lab, but if somebody suddenly tells you, okay, I'm sending you 500 samples a week, then you probably find out you don't have the throughput. Yeah, and so you have to develop a platform where you can actually have the throughput, and so we have to essentially redo some of the work. And if you use mass spec as a measurement, which you can get the throughput and you have the accuracy, but then there may be some slight changes to how we would go about this. So we're working with them in order to establish how we would go about using measurements that would allow for high throughput, so that you can really use this in a commercial setting. Yeah. It's being worked upon. I mean, it's like I don't have an exact timeline for this yet, but things are in progress. I mean, that IP has been licensed. There are plans for basically, well, there are plans for getting to the next stage there. But as you know, right now it's very hard to predict. A lot of it. Some of it depends upon venture capital funding, and with COVID-19 going on right now, everything is very hard to predict where things are going, unfortunately. So I can't put a timeline on it, but it is certainly something that's in progress. Okay.
Yeah. Okay. Thank you for that. I would like to also just ask about this a publication that you referenced in this latest study. Published just recently, and this was a publication that was related to the metabolic profile of women, where a single set of metabolites was able to predict if a mother would be in high or a low risk category of having a child with autism. I think it was like an 80 to 90% accuracy to fall into either of those two groups. So I guess, given your research, is your goal also to create a test for women where they could know whether they're in a high or low risk category before they become pregnant, and hopefully find ways to prep their bodies if that happens to be the case?
That's certainly a long-term goal. Yeah, I mean, we do quite a bit of work right now on by looking at mothers. Yeah, and the paper you're referring to, we had samples from mothers during pregnancy. Yeah, and which is fantastic for because it tells you really what's going on as the child develops, but it's very difficult to run as a study. I mean, our collaborators, they had to they had identified mothers who already had one child with autism, therefore they were at a higher risk that the next pregnancy the child might have an ASD diagnosis. But you had to follow up with them years later to figure out, so does your child now, is it typically developing or do they have autism diagnosis? Yeah, but we are looking like that, but was that paper, but we are looking at, for example, we have some studies on mothers a few years after birth. Or when we look at the metabolic profiles, what are we seeing? Yeah, and so these are certainly things that we're currently working on as well. Yeah, because it is, I mean, ultimately, yes, I haven't seen anything that will tell you that, oh, your child will definitely have autism. That doesn't work, okay? But if you're at a higher increased risk versus an average risk, I think that's an important thing to know as well. Yeah, and it is certainly of interest to us. For our side, I mean, the techniques we're using are very similar. It's not much different from the work we do on the children, but of course, the interpretation is very different and how you collect the data.
Yeah, all of this is like super critical research. I mean, and just to recap on some of the things that we have discussed. I mean, you know, the objective of the goal of some of these research would be to diagnose children much earlier if possible, and to have a reliable way also to measure. I guess the efficacy of treatments. Do you think that would also be within the scope of the tool to have kind of an objective way to go and look at how efficacious a certain treatment is?
Absolutely. I mean, we actually have one paper on that, and the MTT study was a second one. Yeah, that because we want. I mean, what you want to, or what makes this progress in this field challenging, is that there are no interventions where you give an intervention and then suddenly the kid feels so much better within a few days that it's clear cut. Yeah, sometimes you're looking at minor changes, or you're looking at changes that happen over very long periods of time. Yeah, and so if it's something that is not just based upon what did the parent survey say right after the intervention versus 3 weeks later, when you have trouble recalling exactly, well, is this an 8 or 9 on my 10-point scale here for certain behavior? So if you have some objective measurements that help you with saying, look, this has improved, and that's usually an indicator for certain behaviors, I think that would be very helpful. So what we're doing is that I don't think it's going to replace a diagnostic or anything, but it's really more what you may call a companion diagnostic tool. Yeah, that you use this in addition to the observations, but it is one thing that's easily quantifiable, and we say it's not just based upon recall where you may have recall bias.
Mm-hmm. Yeah, this research, doctor, and I guess we're closing on the top of the hour here, but I guess your research, one thing that it does and does very clearly, you know, along with all the clinical trials, is to really highlight the fact that autism is medical and it's treatable. So that's really, I think, a very, very important message, and also that, you know, there are some biomarkers that can, you know, clearly and objectively, you know, give us an indication of progress, which is fantastic. Before we go, doctor, is there anything else, anything specific that you would like to communicate to parents, doctors, and researchers in the autism community?
I mean, nothing super specific other than that, saying this is, I mean, unfortunately, it's a very challenging, challenging condition. It's also for a scientist, not easy to deal with because of the heterogeneity and everything involved. But we and many other people are actively working on that. And I mean, the goal is that ultimately, hopefully, we'll come up with procedures that make everybody's lives better in the long run. But it's clearly, it will take a while. I mean, I think there are some promising things out there that needs to be validated and that hopefully become available in the near future. But there's also a lot more work to be done in the long run. But those are things that some people are actively working on. So.
Yep. Yeah. Thank you so much for your time, Dr. Hahn. I mean, this is tremendous, and we do commend you for all your work, and it's very much needed, and it's actually, as you know, very urgent. I mean, given the state of the the pandemic at this stage. So I really hope that you give us another interview when we have more information about the tools that you're developing, and we wish you luck, and please, we're here to help in any way we can. So thank you for your time.
Well, thank you for having me. So
thank you, and see you next time.
Yeah, bye-bye.